I had the opportunity to meet Kai Miller probably about 11 years ago or so at Stanford. You don't meet too many people who have 2 PhDs. So when I was visiting there, I was struck by him and his commitment to what he was interested in as a mid-year or even junior resident. And it struck me. And, you know, again, in the recruitment mode, I didn't have a position, but I gently asked whether or not Kai would ever think about the Midwest. He frankly said, no, there's no way. He doesn't like cold weather and Seattle and rain. And he gave me about 10 reasons why he would never come to Rochester. And I said, oh, that's too bad because the future is now. In any event, 2-3 years later, out of the blue, I sort of get an email from Kai saying, you know, I'm going to the meeting, would I be interested in meeting up with him? And he said, you know, you talked to me a few years ago, I had a job, it didn't work out, whatever. I might be open to a possibility, and I didn't have a job, I didn't have an FTE, whatever. And he said, and by the way, I need a job in like 3 weeks. Mayo works slowly. 3 weeks under the administration before Bold Forward, 3 weeks, I mean, to get an FTE is about a year process. 3 weeks. And by the way, my wife needs a job. All right, so here's the neural networks, Greg. So I call up Greg and I sort of said, well, let's see what we could do because the future is now. The vision, the networks. Here's the guy, here's the woman, here's the team to join all of these other pieces. So Greg, Prakash, and I knocked on Greg Gore's door. And we said, hello, you know, we're here. And there was a lot of preparation before all this, but this was to get sort of a position for Kai, which I had separately done, but then for Dora, without American grants and all of this other stuff, kind of to believe in the vision, and it happened. But now just, that's Kai and Dora, and it's all worked out beautifully, impeccably. Part of the vision with Kai, though, is they were going to be spending the year in the Netherlands on the Van Wagenen, which is the most prestigious scholarship in neurosurgery. We had never had one. The University of Minnesota had several. How could we kind of compete with the University of Minnesota? Nathan's staff. Nathan would be the mentor to Kai. And I'd followed Nathan's career because my lab efforts were with Tony Windebank, and I think Tony was your biggest cheerleader ever since you were in nursery school. So Nathan and I have this old history, even he doesn't even know about it. But now our kids are friends, so that makes it all relevant. But the point is we wanted Kai to come to Mayo before he came here with a K award. So I flew him at my own dime to interview because I thought, gosh, if I could get him before the committee for the K-12, he would blow their socks away. So Lisa Fabian pulled all the tricks out of the trade because international travel was against Mayo policies for someone who didn't work here yet, but he was a Mayo scholar and all this kind of stuff. But the point was dogged determination by Greg, Nathan behind the scenes, and then Kai and Dora just doing their everyday stuff. Network, 6 degrees of separation. I'm so proud of everyone. So Kai and Nathan, you've got the floor. Make us proud.
Thank you. So that was Lou Gehrig. At the time, he was probably the most famous baseball player in America. And he developed ALS. So he was noted to have worsening batting average during the 1938 season. He actually came to Mayo Clinic and was given the diagnosis of ALS in June of 1939. He retired in July, gave that speech, and died 2 years later from this disease. He, while he was in Rochester, anecdotally, he went and visited people. He was at the local Piggly Wiggly visiting with a former teammate of his who was a butcher at the Piggly Wiggly, and a snapshot was taken in a little article in the Post Bulletin, which is still around here. So as Rob said, I'm Nathan Staff. I'm a neurologist, and I direct the Mayo ALS Center here at Mayo Clinic. And Kai and I are talking together about the development of a patient-centered brain-computer interface platform for people living with ALS. We initially had 2 separate talks, but as we were developing the talks, we felt that it actually would make more sense to do it together. I'll start it, he'll do the middle, I'll bookend it on the end, and I think it does a nice job of also kind of showing off the collaboration between neurology and neurosurgery. So let's level set. So what is ALS? It's amyotrophic lateral sclerosis. It's a fatal, progressive, paralytic disease. It's caused by neurodegeneration of the motor neurons that live in the brain and in the spinal cord. And about 20% of people will also get a frontotemporal dementia associated with it. It's a rare disease, but actually the lifetime risk is 1 in 300, so it's not quite as rare as one might expect. It's just there aren't that many people around living with it because of how quickly they die from it. 90% of people have sporadic disease. We don't know why they get it. 10% have genetic disorder, typically an autosomal disease. There's roughly 30 genes that can cause ALS. Symptom onset to death, 3 to 5 years, causes respiratory failure eventually. It's quite variable phenotype. Some people start with speech problems, some people weak hand, weak leg, and we really don't have any effective therapies. This is a person with ALS showing the hallmark atrophy of muscles. That's the result of the neurodegeneration of the spinal motor neurons. Again, happens in the face, arms, and legs. Number of people, notable people have had ALS. I already mentioned Lou Gehrig. Probably know of Stephen Hawking. The founder, developer of SpongeBob SquarePants, Stephen Hillenburg, had ALS. Some musicians, Charles Mingus, Roberta Flack more recently. Steve Gleason, a football player who's been very active in the ALS community. And more recently, Eric Dane, who's an actor from Grey's Anatomy, has come forward and talked about his experience with the diagnosis. So I think it's helpful to kind of think about a specific patient. So this is an example of a patient that I've seen in clinic quite recently. MK, a 50-year-old man who has 3 small children. He developed left-hand weakness in 2019, got a diagnosis roughly a year later, which is typical. Became walker dependent, needed a walker to get around that year, got speech disturbance the year later, needed a feeding tube because he could no longer swallow effectively and maintain nutrition, became wheelchair dependent that same year, and by the end of that year he was dependent for eye gaze technology in order to communicate with loved ones, ask for things, control the environment. And I'll talk more about that later. And then became ventilator dependent, went ahead and had a tracheostomy and ventilator put in, primarily because he still had the small children, wanted to see them grow up, had plenty of reasons that he could articulate that he wanted to keep living and have a full life despite all the limitations that he had. The eye gaze dependency, so again, had to basically look on a screen to find words and letters and spell things in order to communicate, interact with his environment. Very exhausting. His eyes would get very fatigued and sore every day. Really wish that he could have obviously not used that. Really could only use it in the house. So most of the eye gaze technology does not work well out in the sunlight, despite the umbrellas that they would try to do to get in the way of the sunlight. And then ultimately, with this disease, the eyelids will get weak and the eyes will close. So then the eye gaze technology doesn't really work at all. So pointing out that there's really an unmet need in ALS care. We have this progressive loss of communication, loss of control of their environment, no autonomy, no agency. And the solutions that we currently have are inadequate. So, talking about brain-computer interfaces. So for the purposes of this talk, a BCI is a technology that enables direct communication between the brain and an external device by translating that neural signal into digital signals. So from a cartoon standpoint, we take a signal from the brain activity wave, and then that gets decoded via computational techniques. And then that signal can be sent out, and it could either go back into the brain to stimulate for therapeutic purposes, or it can go out and control some external device, be that a robotic arm, a cursor on a screen, or a speech device. And so we really have a vision for a patient-centered BCI program for ALS here at Mayo. What does that mean? So we're developing focus groups and speaking with patients. We're developing questionnaires and surveys to really understand what their needs are. And as in every walk of life, there's many different types of personalities and needs for patients who have ALS. And we need a high fidelity BCI that's superior to the current eye gaze technology. We need something that goes faster than they can do with their eyes. It needs to be able to be trained very quickly and integrate with our current communication system and home control system. So there's a lot of assistive technology that already exists that can be controlled by eye gaze. We want to control that through the BCI systems. And we want that to be able to be used not only at home, but also out in the community, so they can go out and be outside and interact in the real world. And part of that means that they need to have minimal technical support requirements. So we don't want to have a technician that needs to come out every day to tune the system, to troubleshoot things that are going wrong. It needs to work out of the box and work on a daily basis with minimal interruptions. And so this kind of leads to some problems that are becoming evident in the field of brain-computer interfaces, particularly for implanted ones, that there can be some challenges trying to conflate the idea of somebody who's actually a research participant versus what is the therapeutic intent for these people who are volunteering for the studies. Many of the studies that exist now don't allow for independent at-home use by the participants. And there's often lack of clarity about what the real risks of the implanted devices are and how long is that device going to work inside the brain for this person. And they've had a lot of splash with some very, phenomenal results. But really what we think we need to have is a durable functional prosthetic that has clear neuroscientific goals through the program development, and something that can have a standardized workflow and interoperable interchanges so it can function well within the current systems. And so we're really looking for transparent communication with the patient and the public about brain-computer interfaces, really needing open access at home, long-term support. So again, when these devices are implanted for life, and this life may be decades, what's the support, technical support that the companies and the programs are going to offer for that? And we want it to be scalable and useful for the general community. So we've seen a bit about this already, but there are many methods to measure the brain activity which could then be used to feed into a BCI machine. We've talked about TMS, kind of more macro scales of the EEG, which is the standard in neurology care. We can implant electrodes and do stereo EEGs to get population recordings from groups of neurons, as well as deep brain stimulation. Electrocorticography, so placing electrodes on top of the brain to get, again, population data. Or recording from single units, single neurons within the cortex or deeper in the brain, getting spike information from those neurons. And I think it's important to recognize that these different scales of electrical activity and brain activity really come with different considerations when thinking about developing BCI, and that's what Dr. Miller is going to talk kind of more in depth about in the next few slides. So to come back to the idea of a patient-centric brain-computer interface, we have to be able to choose the brain signals that scale and allow for a robust control of the BCI system, ease of learning of the BCI system, portability so that people can take it where they need to go, long-term signal stability so that electrode that's implanted continues to give a signal that can be used by the computational device, and scalable so it can integrate both into the existing assistive technologies but also integrating within the clinical practice. And I'll hand it off to my colleague, Dr. Miller.
Thank you, Dr. Staff. So what I'd like to tell you about is a few of the principles that we think are needed to have clinically relevant, patient-centered brain-computer interfacing. There are these systems that we need to build, regardless of the one that we're going to show you or some of the ones that you may have seen in the news, are all quite complicated, and there are a lot of relevant considerations. And so what we'd like to do is just give you a flavor of that by going into a few of them and describing them in a way that hopefully will give you some intuition for what the salient issues are. And I'd just like to stress that what these represent are a couple of key lines of research and inquiry that we have amongst a larger milieu of studies that are going on, not just with the two of us, but also with other partners that we have at Mayo Clinic and with collaborators around the world. So there are a number of different ways that we can measure neural activity, and we're going to focus on the electrical ones. Dr. Staff has already spoken a little bit about the different kinds of devices that are out there. We have electrodes that go into the depths of the brain and have electrode contacts that are essentially metal surfaces that are a few square millimeters of exposed surface that we can measure the voltage from. And we can also have microelectrodes, and these measure from a much smaller surface of exposed metal, and they're capturing more like single cells. And then we also have surface pads, which are electrodes that are also averaging over about 500,000 neurons from the surface of the brain. And there are a number of different biomarkers we can extract from these that are, in some cases, very well characterized, and others, learning them is an ongoing process. And some of these might be things that you've heard of, like rhythms in the brain or oscillations. And I'm just showing traces in red to indicate the types of changes that we see by eye in these when there's an engaged state, meaning somebody is moving part of their body, trying to speak, trying to engage with a memory, that kind of thing. We can also, if we give patients stimuli or we look at the time that they engage in some action, see deflections in the raw voltage trace that we measure, and we can see this at small scales at the size of 1 or 2 cells, and we can also see this at the very large scale from outside of the head. There's something called broadband changes, and this was, for me, this was the main topic of my physics thesis, was discovering and exploring these. And then there are action potentials, and action potentials are really the outputs of single cells in the brain or single neurons. And this dominates a lot of what we read about in the news and what you read about with devices. And for us, it's important to keep in mind that each potential feature that we measure reflects a different aspect of the brain's computation. Each of these signals is going to be best captured at a different set of scales, so spatial scales, right, with different sizes of electrodes. And individual brain-computer interface constructs may benefit from a synthesis of these. And our job as scientists and clinicians who are trying to make something that's patient-centered is to look at these signals and try to understand what are some benefits and what are some limitations of each of these. So when we think about these oscillations, something that's good about them is they're present over very large regions of the brain, so much so that we can pick up on them outside the head, but they can be functionally nonspecific. So if you see a particular pattern, that can reflect something as wide as moving let's say the hand or the face, and sometimes from outside the head you can't pick these up. But there's also a lot of overlap so that, you know, if you move, let's say, your thumb or other fingers, everything is active within a whole domain, whether you're looking at one finger or another. We can also look at these event-locked deflections. The benefits of these are temporarily very precise. A limitation would be they're inherently supervised. You have to know when an event's happened to see if there's been a voltage deflection that followed it. There's these what we call broadband spectral changes. These are functionally very, very precise. They reflect populations of neurons, and I'm going to go into these quite a bit. A limitation going back many years has been that because these signals have very specific statistical properties, which I'll show you, it really required high-fidelity instrumentation to capture them, although there's now hardware instrumentation that's implantable in the body, some of which has been developed actually by Dr. Dennison, who's our keynote speaker tonight, that allows us to capture these. And then there's action potentials, which I talked about earlier. You hear these talked about spikes, single cell activity. So in a way, these are the finest resolution of the brain's computation, But really, they're stochastic. So when you look at any individual cell, at least as much as we can characterize them, it has some randomized properties to it. And they don't have great statistical reliability on their own or particularly good temporal resolution. And I'll show you what I mean pictorially in a moment. So this is an example of a microelectrode recording array. So these are measuring single cells. And they measure the voltage like you see here. We capture small segments of the raw voltage, and we then will filter these signals and we'll capture these events that you see here. These are called action potential times. And what we're doing when we capture these events is we're separating out what we call background field potentials, or background activity that represents all of the inputs that are coming to individual neurons, but not that discrete output that we capture with action potentials. And if we want to do something like control a brain-computer interface with this, and this is a lot about what you read about in the news, really you must measure from many of these at once, and your device has to be able to record from hundreds of channels at once and extract these single events and combine them in some way that gives you some kind of statistical reliability to control a device. So I'm going to tell you about something that's the exact opposite end of this scale from measuring inside of the brain from that. And that's these larger scale electrodes that we measure primarily and learn from primarily by working with our patients who have epilepsy, which you've already heard some about today. And we have these patients who have seizures. In order to identify where their seizures are starting from, they'll first see our colleagues in neurology, and you've already heard from a couple of these today. And then they'll come to a neurosurgeon like Dr. Van Gumpel, who you'll meet tomorrow, or myself, and we implant these electrodes, and then we monitor patients for several weeks in the hospital and wait until they have seizures. And while we're waiting for them to have seizures, they generously donate their time to do tasks with us. And so you're going to hear me talk about electrocorticography, and these are electrodes from the surface of the brain. It's abbreviated ECOG, or stereo EEG, and these are depth electrodes. And these are the same dimensions, roughly, that we have when you hear about deep brain stimulation electrodes, roughly the same spatial scale. And we want to understand things about the statistical properties of these. And the reason why I put dates on all of this is because what I want you to understand about the role that we have and that we want to have in this BIONIC initiative is a culmination of decades of research that we've personally been doing. So back in 2007, we looked at the changes that we saw in these electrocorticography signals when we had patients stare at a blank screen. And we measure something called the power spectrum from that. And I'm not going to go too much into that for those of you that may be more mathematically oriented in your background. We look at the Fourier transform and calculate the power spectral density. And we can look at, and this is extracted from those voltage traces that we see, and we can look at that state during rest when they're staring at a blank screen. We give them a cue to move their hand, and we get a different power signature here. And what we see when we overlay these two is that if you look at low frequencies, there are these very spatially broad changes in power that are across wide regions of the brain, and they are really narrowly confined to specific frequency ranges. So these are the types of things that you're used to hearing about that we would call brain rhythms or oscillations in the brain or brain waves. But at high frequencies, where you see changes that are spread across a broad range of frequencies that obey something statistically called the power law, we have changes in power across a very wide range of frequencies. And these really look like, to your eye, like random walk changes in the signal. And those have important statistical properties that I'll get to in a moment. But when we looked at this, we thought, well, if you're looking for a process that is inherently stochastic, like action potentials that I showed you, and you want to know what the aggregate sum of all of those action potential events are, this is what it would look like statistically in your signals. And so if we want to get maybe at mapping local activity in the brain, maybe all we need to do is capture this. And so what you're seeing here is the first demonstration of real-time electrical brain mapping from the brain surface with these types of electrodes 18 years ago. And if you look at the hand there, you can see on the screen, and the hand here, a lot more spots now, unfortunately. So the patients there on the left side of the screen. What you're seeing on the right side of the screen is a much younger version of myself back when I knew Dr. Lundstrom, who's in the audience. And this was actually a great year because maybe the best year of my life, because this is when I met Dr. Hermes, who you're going to hear from tomorrow. And the brain activity that you're going to see was plotted on the brain in real time in front of the patient, and that's what you're seeing. And so you could see the representation of this patient's arm and hand in real time. Okay, so we realized that by capturing these broadband spectral changes, we could map very local function in the brain. And now, 16 years later, so a couple of years ago, we said, well, do we do we see the same things with these depth electrodes that we put in the brain? And the reason why we have depth electrodes in the brain now and surface electrodes 18 years ago is there was a large paradigm shift in how we treat epilepsy patients, such that now we primarily have depth electrodes in the brain. And this is the work of now Dr. Michael Jensen, who's part of our team. And we did the same thing where we had a patient stare at a blank screen. And what you're seeing on the just to the left of the brain there is, or the cartoon brain, is a slice of an MRI through the movement area of the brain. And we measure this stereo EEG voltage. And we give them a cue to move their hand. We can look at the signature from their muscles, the electrical activity from their muscles, called EMG at the same time. And we see the same characteristic changes in the power spectrum. And so all we really have to do to map local function, we've now found, is to capture these broadband spectral changes. And that works whether on the surface of the brain or at the depths. And that's important if we want to have a brain-computer interface, because that tells you that we can have a combination of surface and depth electrodes when we do it. And we can immediately map motor areas, as you see here, and sensory areas. And that, for those of you that are neuroscientists, which most people in the audience are, that yellow line is the dividing line of the central sulcus between sensory and motor. And when we statistically model these, we can model, let's say a toy neuron here, and its inputs to generate these field potentials that we get as a series of input action potentials, so not measuring the output of cells, but we measure the inputs to a population of neurons. And these have characteristic changes that they introduce to our neural population. And when we model the time scales of these and the statistical behavior that they should have as those signals make their way from the input part of these cells, called the dendrites, towards the cell body, they have very specific statistical properties we can model mathematically. And when we do this, what we find is that when we increase the activity in neural populations, it mimics what we see in our signals that we've made actual measurements from. And the implication for this for electrodes and for designing brain-computer interfaces is that the size of the population of cells that we're looking at roughly scales with the size of electrode that we have. So it's a great way to interpret neural population activity. And something that we explore, but I'm not going to go into, is how the brain's representation of things like movement changes as you change the size of electrode that you're measuring from. And there's some counterintuitive things that are very important for brain-computer interfaces that come out of that. So we can capture those broadband changes here as a function of time, these high-frequency changes. And these are pictures of a brain. That yellow line is the central sulcus. The green line is the Sylvian fissure. And I'm going to show you activity from the electrocorticography measurement made from a precentral, which means in front of the central sulcus, hand movement electrode that happens to be specific for thumb movement. We measure position of the fingers with data gloves. And we can see those power spectral changes that I showed you before, where we have them as a block. And we can see these low-frequency oscillations and how they come and go in time. And if you compare that yellow trace there to the movement of fingers, you can see that there's a decrease in power with movement, but the temporal fidelity of that is not great. But if we capture those broadband spectral changes, what we see on the brain itself is that the representation is very sparse. It's very specific for specific regions of the brain. And the temporal resolution here is capturing the dynamics of individual finger movements. So this is a great biomarker to drive brain-computer interfaces. And I showed you that for one finger. This is what the electrocorticography grids look like on an X-ray. I always thought that was really cool to see. Of course, we've seen it on the brain. We see it in our interpolated pictures of the brain. And for individual fingers, you see changes corresponding in different brain sites corresponding to individual finger movements. So we can capture the brain's representation with very high temporal fidelity here. And so with the electrocorticography signal, we capture this signal, and it's great. And we need only one channel of electrodes and some online processing to capture that. If you want to use the technique that dominates the news now, you need devices that can capture and amplify the signal from about 100 sites at once and combine those. Okay? And that doesn't mean that that's a bad approach, but it does have its limitations. So using macro electrodes, meaning larger electrodes, there's less sophisticated hardware requirement, and there's less sophisticated translational algorithms. And so if we want to decode brain states with artificial intelligence, and that's something we can do using these different signals. We capture these different signal features. We make them an input layer to a neural network. We apply our artificial intelligence algorithms, and we have end-state effectors that we can then use to control robotic arms and other things, or we can decode perception. And what you're going to see here is how we do that by combining different features to predict what a patient's seeing. A patient's seeing pictures of faces and houses. We apply artificial intelligence algorithms to a feature space that we've created out of those signals. And you're going to see the real-time prediction of our algorithms to decoding perception in real time. Okay? And this black line that you see on the bottom right is the instantaneous brain state. And I'm showing you a three-dimensional feature space. But of course, our feature space is, well, it's up to as many salient dimensions as we have. But usually, for an array of electrodes that we'll put in, we're looking at something like a 200 to 300 dimensional space, with about 20 of those dimensions being relevant. And we can detect the onset of these visual responses with millisecond accuracy, and we're very good at it, right? So the onset that you see up there, that's not labeling the pictures face and house. That's the output prediction of the algorithm, okay? And this is optimal decoding. And so that requires retraining of that decoding algorithm on a case-by-case basis, okay? So all of these beautiful demonstrations that you see of people recreating speech using either these macro-scale electrodes or single units, they're retraining these day by day. It's really hard to bring that to clinic. It makes for great news stories. It makes for great ways to push your IPO, but it's not the kind of thing that I want to use to help patients. Another thing that we can look at is we can see, instead of creating very sophisticated algorithms, and we're very good at doing that, we can create some things that are less sophisticated but give the patients more control using something called operant conditioning. And here we're going back to these same power spectra, and we're going to have patients control cursors using thought, okay? And if we look at hand and tongue representation, kind of like I showed you before with fingers, we see they're not overlapping. If we have patients imagine moving their hands and tongue, we see that the overlap between tongue imagery and hand imagery with overt movement is very similar, right? This overlap is very high. And so we can use imagining moving your hand and tongue to drive cursors left and right. And all we do is we take that high frequency power from two different sites and couple it to left and right movement and up and down movement of a cursor on a screen. This, of course, isn't great compared to a lot of stuff you see in the news, but this is after 10 minutes of training in our epilepsy monitoring unit. Patients control cursors in 2 dimensions. And the important part about this is not that they can control a cursor. The important part about this is that we're operantly conditioning these signals. And operantly conditioning means an individual learns how to control the signal. So if we track the power from that one brain site in that high frequency as a function of time, we can look at upper and lower targets. And we see that over just 10 minutes, patients can control the power in that one site, so they have volitional control over it. So they're learning the mapping. Our algorithms are not decoding it in any sophisticated way. So if the activity of the brain changes, if these signals change slightly with attentional state or arousal or medications, then patients can adjust to that. And we can do the same thing with stereo EEG brain-computer interfaces here at Mayo. And we've had a program going for the last 5 years doing that. This is just an example of this. And this is with stereo EEG, so it's showing that with these depth electrodes, we can do the same thing. Okay? And we see the same process of learning. This is, again, Dr. Jensen's thesis project with rapid operant conditioning control of individual sites. Okay? And the mapping approach here is that individual signals using sophisticated machine learning algorithms, but channel by channel, and I guess I should said sophisticated, but it's not really that sophisticated. It's extracting these broadband signals and coupling those directly to the individual components of the end effector creates robust systems that are where the patient is able to relearn the mapping, and this is very different from neural network approaches. So we do both. Neural network approaches are great for decoding. When you're trying to optimize things for a demonstration or you're decoding perception, it's great. For having robust control over days and months, maybe not the best thing. So these are all some of the different things that we think about. I've just shown you 2 of the principles that we're exploring to give you a flavor of what we do. And these are the kinds of problems that we need to integrate into our platform. And our platform is this. It's a 32-channel sensing and stimulation device funded by the NIH, combining an open source software platform called BCI 2000 with a commercially available device. We're going to use this for back-end control into assistive devices so patients don't have to learn something new. It's just that now they're going to combine eye control with brain control until they don't have eye control anymore. We're 2.5 years into preclinical testing with this. We know our signals are stable over the course of years. And Dr. Worrell showed you that earlier. And we have this integrated with the cloud, and we're hoping to have this working within the Mayo platform using Dr. Staff's team.
Thank you, Kai. And so coming back to MK, our patient who's been using eye gaze, what does that person's future look like? Well, starting the discussion of BCI implantation early after diagnosis. Not everybody's going to want to do this, but I think we'll start to include that as we do with other things that we do in our ALS clinic. Implant, perhaps, at the time of dysarthria, once we've really done a thorough informed consent on this. And then they're able to use the BCI for communication autonomy and improved quality of life. And so just to get back to Rob's, this was 2018, Greg and Kai and I at a somewhat known coffee shop in town. And then that is us in Utrecht with Dr. Dennison as well and Dr. Hermes. And this was more recently at Forager. Just quick run through the financial disclosures. I wanted to point out two funding sources that have been very important, the UO1 that Kai, Greg, and Peter Brunner have, as well as the Chen Chao and Chrissy Chen Institute that have funded Kai and I as scholars. Thank you.